So as Michelle said, my name is Kalina. I'm a scientist in Richard Wade Martin's lab, who I'm sure most of you have met as well. I am advised by Brent Ryan and Richard, and today I'm going to talk about the project that I'm doing alongside Richard, Michelle, Ludo, Caroline, who spoke earlier. We're all part of the Oxford GSK collaboration, and we're part of the Institute, the IMCM of Molecular and Computational Medicine. And in this project, we're trying to address sporadic Parkinson's disease, which I'll explain a bit more in my next few slides. But first, let's just start. I'm going to talk a lot about cell biology, so I'm going to try and make this a little bit better to explain. So keeping a cell happy is like keeping a home happy. So what are the things you need to keep a home happy? You need light, energy. You need good, clean water. You need good waste management, so recycling, remembering to take the bin out on bin day. And you also need, obviously, the infrastructure of your house itself. And in many ways, the cell needs these same things as well. So the cell needs energy. It gets that from an organelle called the mitochondria. Managing the water and the liquid in our body via the blood and the CSF. We're also, waste management in our cells are managed by an organelle called the lysosome. So it's in charge of breaking down things we don't want in the body and recycling things that can be used for other processes. And then obviously the cell architecture itself, so that's the cell infrastructure. We need it to be a certain way for it to function. And so in the OPDC and in Richard's lab, we are looking at these specific organelles. We're looking at CSF, we're looking at blood, mitochondria, lysosomes, and the actual cell morphology, so the shape and how they connect to other cells. So I'll be referring to specifically the energy makers of the body, of the cell, which is the mitochondria, and also the waste management system, which is the lysosome, in my talk. So, we're here today discussing everything about Parkinson's disease, and in our project, we're hoping to address the three challenges in our project, three challenges of Parkinson's disease. So the first challenge we know is that we know that there is a loss of these brain cells in a specific area of the brain responsible for movement, which speakers have mentioned earlier. But what we don't know is why are these specific cells being lost, and why, what are the biological mechanisms for this? So this project is trying to look at this. In general, when doing Parkinson's disease, we can look at familial types of Parkinson's disease, which is about 5 to 10% of people will have a familial type. So this is a big genetic driver of Parkinson's disease. So you'll hear things like a GBA mutation, LOCK2, etc. Or you could have a sporadic Parkinson's disease. So this is something where there's not a big genetic driver. There could be small little genetic drivers that add up, but not a massive genetic driver. And so in our project, we're very interested in the sporadic Parkinson's disease. And we're interested in specifically the energy makers and the waste management system. So the mitochondria and the lysosomes. And what is interesting about this project is it's based on work that Michelle has done previously with someone called Michael Lawton, and they've actually looked at the tracking Parkinson's cohort and the discovery cohort, and they've clinically subtyped the patients in that cohort into 4 clusters. So I'll discuss that a bit more later on, but what that means is they've looked at the symptoms of the patients, and they think that the patients fit into 4 distinct clusters. And what's exciting for me is to try and find out, is there a biological underpinning for what they're seeing in the clinic? And I go about doing this by looking at assays targeting the mitochondria, which is energy production, and assays targeting the lysosome, which is that waste management system. And how I'm going to do that specifically, and how I am doing that specifically, is I'm looking at fibroblasts, which are skin cells. So when you give your skin biopsies, if people have given skin biopsies, thank you so much, if you've given those skin biopsies, I get to take that, someone kindly up a hill isolates it into something called a fibroblast, which is just a purified skin cell, and I get to do experiments on that. And I'm going to report back a few of those experiments. So this is just a description of Michelle's work with Michael. And so as you can see, this describes the 4 clusters of sporadic Parkinson's disease across 2 cohorts. The tracking cohort had 1,601 patients, and the discovery cohort had 944 patients. And I want to say thank you so much, because none of this research today could be done without the help of patients. And so what we think the clinically is telling us is, in these 4 clusters, the first cluster has a fast motor progression, so the motor aspects develop very quickly. The second cluster is a bit more of a mild cluster. The third one is a more severe cluster in the sense of cognition and psychological symptoms, and the fourth one has slower progression of those motor symptoms. And also part of my project, but not done by myself, done by someone else in our group, they're actually going to look at the second and the third cluster, and they're going to take these skin cells that I'm working with, and they're making them into neurons, specifically those dopamine-producing neurons in the brain. So they're making them using iPSCs into neurons, and then they're going to do further investigation comparing this milder cluster to the more severe cluster to see a little bit more in a neuron-specific model what's happening. Okay, so in my project, as I said, I'm very much interested in the energy producers, the mitochondria, and the waste management system, so the lysosomes. And why am I specifically interested in this? A lot of papers have reinforced the idea that the mitochondria and the lysosome is very important in Parkinson's disease. We think possibly the reason why these brain cells die first, the ones responsible for motor, specifically in this the dopaminergic, the substantia nigra region, we think it's because they need so much energy, they're the first to die when they don't get enough energy. So we think that's why we're targeting the mitochondria. With the lysosome, especially a few speakers spoke about this earlier, when we say there's a aggregation of a specific protein called alpha-synuclein, and we think that it's not being removed, so it's normal to go and then look at the removal system in the cell. And I'm doing this by looking at specifically the shape of mitochondria. So when these mitochondria are not happy, their shape changes. We're looking at oxidative stress. So we all know about antioxidants and how important that is to keep your body happy, and so maybe we're not getting enough antioxidants in that mitochondria for many different reasons, and that means that these bad reactive oxygen species but more, and then we have oxidative stress, and then also energy production, so the actual quality of the energy that is being produced. So I have different experimental assays that I observe this in. Then the waste management system, the lysosomes, I'm specifically interested in enzymes. So enzymes are things that speed up a biological process. So for example, if you were making a cake and you needed to mix the batter, you could use a whisk and it would mix the batter real nice. But if you use an electrical beater, it's going to whisk it much faster, much smoother, much cleaner. So that's why you need those enzymes. So if you don't have the right amount of enzymes being made, you may not be able to do the function of that lysosome. And so I'm specifically interested in an enzyme that is responsible for breaking that down. So one that's responsible for breaking down fatty stuff is GCase, and then just normal, what we call proteolytic enzymes, which is for proteins. I'm also very interested in the pH in the cells. So I say here acidic is happy and alkaline is sad. So our lysosomes, the specific waste management system, they need to be acidic. So they need to have a very low pH. If this pH is too high, then those enzymes that I spoke about, they're not able to do their job. So we need the pH in those cells to be capped quite low. And lastly, I'm also looking at the actual recycling capability of these organelles, the lysosomes. So we know that it doesn't just break down products, it also makes it into new ones by recycling it. Okay, so how do we do this in our lab? So we get the skin cells and we grow them over a few weeks. And so I've got some pictures to aid along the wet lab process. So as you can see, that's me and a colleague looking down the microscope and making sure the cells are healthy and happy. We culture them in this incubator, where it makes, in this incubator, it keeps the temperature at the right levels, and so the cells grow very healthy and happy. And then we do something, we say we add dye. This is something we say a lot in our lab because we do a lot of imaging experiments. And dyes are just something you add to a cell or a tissue sample that the cell will take it in, break it down, and release a fluorescence that the microscope is able to monitor. So there are usually these bright, like those bright red structures and the green structures in the cell. So the red structures is actually the mitochondrial networks in that cell, and the green bits are just the cell itself, just to see the shape of the cell. And so we image this using what we call our most valuable player of the lab, the Phoenix. And this is our imaging system, which is able to do really high content level imaging very quickly. And then we take these beautiful images and we analyze them and try to make sense of the data that we get through graphs, which I'm going to show you in a bit. Okay, so the first part of my project was actually quality checking all of the skin cells that I had, that we were able to get from the OPDC cohort. And so this was a process that involved growing up these cells and then storing them down in smaller vials so they can be used for the next 2 years, which is how long the data acquisition part of this project is meant to be. And then we do a lot of tests to determine things like how fast they grow, so we understand how to put them in specific places in the experiments, because we want the cells that grow really fast to all be together and the cells that grow maybe a little bit slower to be together. So we're only comparing what we think are biological differences and not technical differences. The next part is something we call we say we have to establish assay windows. So assays are just these experiments. So the experiments that we're looking at enzyme activity, the experiments that we're looking at the shape of these mitochondria, they're all different assays. And to set these experimental windows in the sporadic disease, we actually used familial Parkinson's disease, which we know are these major genetic drivers that target very specific things in the cell. So the GBA-N370S, for example, is targeting the lysosome, the waste management system. And so you're going to see the most extreme changes between healthy volunteers and people with the GBA-N370S mutation in the lysosome. With the LRRK2, this is more targeting the mitochondria. So you're going to see the most extreme changes between healthy volunteers and people who have the LRRK2 G2019S mutation. So we think that that's going to set those two extremes, the healthy volunteers and the people with these familial Parkinson's disease. And then someone with a sporadic Parkinson's disease, if they have a lysosomal or mitochondrial change, it will fall somewhere in between there. So we want to make sure that we're setting the right parameters for when we do this big 91-line sporadic screen. And so we're establishing this in these familial Parkinson's disease types first to set those windows, and I'm going to be telling you a bit more about the data that we got from that. So I'm only going to present the lysosomal data. So this is the lysosomal data in either the familial Parkinson's disease GBA type or the LRRK2 type. And remember, this is the one that's all about waste and recycling, right? And so I'm going to be presenting the enzyme. So remember, this is the thing that helps speed up a process, that specifically the ones that break down fatty substances, the pH of these organelles, and also the proteolytic activity of these organelles, so the lysosomes. So the proteolytic activity is how well does it break down protein, and also measuring the ability for the cell to recycle its substances. And so I'll just dive right into the data. And so what we're seeing here is you're seeing fibroblasts, so skin cells, from patients with a GBA N370S mutation in blue, in the blue graph, and then on the green graph, it's a patient with a LRRK2 G2019S mutation. And there'll be about 4 patients of each type versus an equivalent of healthy volunteers. And what this means is that there's a reduction in this enzyme levels in patients with the GBA N370S mutation, and that's what we're seeing with our fibroblasts. So this is something that has been published quite a lot in the Parkinson's disease literature, so it's good that we're confirming that finding. We do not see a change with the LRRK2s, but we're already using that more for a mitochondrial phenotype. And this little guy on the side here, where it says it's gold, it's CBE. These are our chemical controls, which we'll usually have in an experiment, where this is something that specifically inhibits this enzyme. So we use it as a control to make sure that if the CBE is doing its job of inhibiting the enzyme, that the experiment works, and we can trust the findings we see in this familial Parkinson's disease type. Moving right along, this is the lysosomal pH data. Again, we only see a change in the GBA type and not the LRRK2 type. This is understandable because the GBAs are specifically affecting the lysosomes, and we see that it's increasing the pH. And remember I said the lysosomes are happy when they're acidic. So if you're increasing the pH, you're making it less acidic, and so those enzymes are not doing the job that it's meant to be doing. And the images just is what I use to get the graph. So the images, those little tiny spots, are what we measure. We measure how bright it is, we measure how many they are, and we take those little tiny spots and we get numbers from it, and that's how we get the graphs. So this is how well does your lysosome break down specifically proteins, because we know alpha-synuclein is a protein, so we're very interested in how it breaks down proteins. And this is a specific assay that is looking at that. Again, it's the waste management system assay, and so you see those tiny golden spots. That's actually the dye fluorescing when it's in the lysosome, and how bright it is means how active those lysosomes are. How many they are is also something that we take into consideration. The size of them we're able to see with our machinery, and you can see this CQ is just a positive control, so it's something we know for sure decreases this proteolytic activity, and we've established that in this assay. And then we see that the GBAs decrease the proteolytic activity, and so there's something about these GBA and 370S patient lines that have a decreased capacity for breaking down proteins. And interestingly, the LOCK2 lines have an increased proteolytic activity. But in general, we think this is just a dysfunction. So we just think anything on either side is not really a good thing. The LOCK2 could be compensatory for something else. And so this was very interesting to see those lysosomal changes in the petri dish. And lastly, we're looking at how something called LAMP2, which helps us understand how well a cell recycles its product. And with the LAMP2, we see that there's less lysosomes stained with this dye that gives us an idea about how well recycling is happening in the patient lines with GBA N370S. And so that means that something is happening in those lines to affect the lysosomal autophagy system, the recycling system, and therapy should target that to try and rectify it. So, in conclusion, using these fibroblasts from patients with Parkinson's who have familial PD, we were able to determine the right times. So how long should the cells be on the flask before we experiment on them? How long a certain fluorescent dye needs to be on a cell for it to get a good measurement. And so we were able to use these familial types to set our timings for our target assays for the energy production and the waste management in the sporadic patients with Parkinson's lines. And we're going to be screening 91 of these lines. We're obviously not going to do them all at one go, so we need to, we separate them into, we're thinking about sets of 30, and those are really big experiments, so we want to make sure that we have all those timings really tight, and also we want to know exactly how to space them out. And that's what I'll be spending my next 15 months doing. Okay, so thank you to Richard and Michelle and Ludo and David and Caroline, all in our IMCM group, and most importantly to all the patients and healthy donors. We couldn't be able to do this research without you, so thank you.